Namaskar, warm welcome to one and all. This is Part 6, Chapter 8, Unit 3. The chapter is Cell, the Unit of Life. In this Part 6, we have to study regarding microbodies, different type of microbodies, detail of nucleus, chromosome, different type of chromosome structure, and then in the last we will see special type of chromosome, that is 𝕆𝕆𝕋 chromosome and polytene chromosome. This is Part 6, Chapter 8. In this part, first we have to discuss microbodies. What are microbodies? These are the bodies found in cytoplasm, single membrane that is found which encloses the content of microbody, and these are present in cytoplasm. These are present both in plant and animal cell, and these organelles mainly involved, involved in oxidation reaction. That is, oxidation reaction takes place mainly in mitochondria, but oxidation reactions takes place in microbodies also. So you have to say that this is that is cell, this is cytoplasm, this is the single membrane structure which we may call that is microbody, and in microbodies mainly there is oxidation reaction. No doubt oxidation reaction takes place in mitochondria mainly. So three type of microbodies we have to discuss. One is peroxisome, very important one. Peroxisome, very important for examination point of view. Also called as uricosome. They contain enzymes for peroxide metabolism in animal cell. Peroxide metabolism in animal cell. They contain, that is, peroxide synthesis as well as peroxide destroying enzyme, which is very interesting one. That is, contains enzymes for peroxide synthesis as well as, that is, destroying peroxide enzymes are also synthesized, known as, that is, catalase. In plants, very important one, peroxisome in plants. These are concerned with photorespiration. You have to study detail of photorespiration in chapter, that is, photorespiration will be discussed in chapter photosynthesis, also known as glycolate pathway, or also called as, that is, C2 cycle. C2 cycle. In, that is, this cycle there are three organelles. The one organelle, that is chloroplast. Other organelle, that is peroxisome, and the third organelle, that one is mitochondria. All these, that is, these are acting as unit, and these are responsible for, that is, photorespiration. Photorespiration, I repeat, also known as glycolate cycle or pathway, and that is three organelles are associated. One is chloroplast, one is peroxisome, one is mitochondria. And in case of plants which do photosynthesis, especially in C3 plant, that is, photorespiration takes place in peroxisome with association of chloroplast and of mitochondria. Okay. Come to the next now, that is number 2, spherosomes. This is also, all of these are microbodies, single membranous structure, spherical. This is the major site of synthesis of fat and storage of fat. Fat in fatty seeds, they will be synthesized and stored, especially the oil seeds. Their endosperm is having lot of, that is, these are abundant in spherosomes, where, that is, fat will be synthesized and stored. At the time of germination, you all know, that is, this fat will be utilized. So these are filled with hydrolytic enzyme and believed to be, that is, lysosome. These enzyme, that is, will digest the fat, and then they will help in seed germination. These are produced from smooth endoplasmic reticulum. Smooth endoplasmic reticulum. This is, that is, rough endoplasmic reticulum, where the protein will be synthesized. From this is the, that is, ribosomes are there on this. This is the, this is the smooth endoplasmic reticulum. These proteins synthesized here will be transported, and from this smooth ER, that is, these bodies will be bodied off, known as spherosomes. What are glyoxyosome? Again very important one. Glyoxosome, these are found only in plant and fungi. Remember, these are absent in, that is, animals. They contain enzyme for glyoxylate. Remember, this is glyoxylate cycle. This is not glycolate cycle. Confusion is, this is glycolate cycle. No, this is not glycolate cycle. Glycolate cycle that take place there in peroxisome, also known as C2 cycle. What is this? This is glyoxylate cycle through which the fat are converted into carbohydrate. The process is known as gluconeogenesis. This is not glycolysis. Sometimes the question used to come. Glycolysis is different. This is gluconeogenesis. Genesis means what? Formation of glucose. Formation of glucose, that is from fat. Gluconeogenesis. These are found in germinating seeds, especially that is germinating seed which are rich in, that is, fatty acids, fats, such as castor seed, groundnut seed, that is where insoluble lipid food reserve is there. That should be converted into soluble sugar at the time of, that is, germination to provide energy. Animal cannot execute this conversion because they do not have this glycoxylate enzymes. Is it clear? Okay. That was about the microbodies. Now we have to see an important organelle inside cytoplasm, that is nucleus. Nucleus as cell organelle was first discovered by Robert Brown. You can remember the name Robert Brown, as early in 1831 in root cells of orchid. Orchid is the plant. In root cells of, that is, orchid, that was discovered. The material of the nucleus is stained, very important part again. Nucleus, how can we stain the nucleus? We can stain the nucleus with basic dyes, and because it can be colored with basic dyes, that is chromatin, that name was given chromatin. Chromatin means colored body. Fleming did this work. Basic dyes can cover this content of the nucleus, chromatin. It is relatively large organelle controlling all activities of eukaryotic cell. This is known as master of the, that is inside, that is, this is cytoplasm, this is eukaryotic nucleus. All activities of cytoplasm are being controlled by, that is, nucleus. Usually there is only one nucleus per cell. This is one nucleus per cell, but sometimes they have more than one nucleus, as binucleate, two nucleus we have in case of paramecium. Paramecium is having two nucleus. One is larger one is that is smaller one, macro and micro nucleus. In Opelina we have many nucleus, multinucleate, having more than one nucleus. It is very important one. Sometimes the nucleus is there, but nucleus during development, that is, it disappears. Then these cells, they lack nucleus, will be known as anucleate. Anucleate doesn't mean that the nucleus is absent since beginning. Nucleus is lost at the time of maturity. That condition is known as anucleate. And as example found in case of mammalian RBC and sieve tube cells of plant, that is of phloem. Right? Come to the next. You see, this is nucleus. We have to see the structure. This is nucleus here. That is how this is being separated from the cytoplasm. This one is the cytoplasm here. It is separated from rest of the cytoplasm with the help of this nuclear envelope. This is the nuclear envelope, which is made up of, that is, double membrane. That is, it is connected to the cytoplasm through these pores are there. That is, this one is cytoplasm. That is, through these pores it is connected here. Inside nucleus, that this whole content is known as nucleoplasm, karyolymph or nucleoplasm. In karyolymph or nucleoplasm, we have, that is, a body, number may be 1 or 2, that is known as nucleolus. Nucleolus is the site of ribosomal RNA synthesis, and that is one of the main thing known as, that is, chromocenter. This one is, and here that is euchromatin, or right now you can see that this whole thread-like structure is known as, that is, chromatin. And you know that this chromatin we can divide in two parts, two types. One is, that is, heterochromatin, one is, that is, euchromatin. We have to see detail of this in time to come. Come on now. If you see the structure of nucleus, interphase nucleus of a cell, when the cell is not dividing, is highly extended and elaborate nucleoprotein fiber called chromatin and nuclear matrix, and one or more spherical bodies known as nucleoli. The single of this is known as nucleolus. So we have to see one by one. That is nuclear envelope we have to see first. Electron microscope has revealed that the nuclear envelope is present, consists of two parallel membrane, that this is the two parallel membrane like this, and here that is this will separate the nucleus from cytoplasm. This is the outer membrane, this is the inner membrane, and this is the pore that is known as nuclear pore, right? Space between is 10 to 50 nanometer. This very space called perinuclear space. It forms a barrier between materials present inside the nucleus and that of cytoplasm. Outer membrane usually continuous with endoplasmic reticulum. You see here this one is endoplasmic reticulum like this. Endoplasmic reticulum usually in continuation with this, that is outer membrane, is it? And this may be as that is ribosomes, because ribosomes are present here on, that is this is the rough endoplasmic reticulum. Is it clear? That is that bare ribosome. At number of the places nuclear membrane is interrupted, I told you minute pores are there, known as that is nuclear pore. It is formed by fusion of the two membranes, and the nuclear pore that will be the passage through which the movement of RNA and protein molecules will take place in both the direction, either from the that is nucleus or that is from cytoplasm. As per requirement, the things will move from inside to out or out to inside. Okay, come to the next. Now we have to see that is nucleoplasm. Now nucleoplasm, simple thing, that is inside the nucleus. This is that is double membranous structure. Inside the nucleus, what content we have, and everything inside the that is this amorphous liquid, the medium where the other things are dispersed, that is known as nuclear sap or karyolymph. This is transparent, semi-filled with colloidal substance, which fills the nucleus. Inside nucleus, it contains nucleoside, nucleotide, DNA, RNA, protein, enzyme, minerals such as phosphorus, potassium, sodium, calcium, and magnesium. Children, you have to remember that there will be that is DNA duplication will be there inside, and that needs different type of that is raw material. For DNA or RNA synthesis, what is required? Nucleoside and nucleotides are required. Is it? For synthesis, different type of proteins and enzymes are required. All these are present there in that is karyolymph, that is raw material, as well as the things which is required for synthesis of DNA or RNA. Nucleolus, inside this nucleus there is this structure, that is spherical structure. Number may be one or that may be more, that is known as nucleolus. Discovered first by Fontana. The term was given by that is Baumann. Spherical, dense, colloidal, acidophilic body that is present in nucleoplasm. The content of nucleolus is continuous with that of nucleoplasm. There is no membrane. It is in continuation with the that is nucleoplasm, as doesn't have that is membrane. Very important point, as I told you earlier also, in a nucleolus there will be synthesis of ribosomal RNA. This ribosomal RNA, after synthesis, that will move to the cytoplasm, and here it will combine with the protein and will make that is ribosome. So you have to remember that it is the site of RNA synthesis. Larger and more numerous, you see, where more protein is required, more ribosomes will be required, and therefore there will be more and large nucleoli will be present. So nucleoli number and their size, that depends on the requirement, because that is responsible for protein synthesis. The cells which are actively engaged in protein synthesis, they have comparatively more nucleoli, and their size is also larger. Come to the chromatin now. It is very important one, hereditary part of the nucleus. Chromatin, colored, because it can be colored. This is the colored part, chromatin. Interphase nucleus has loose indistinct network of nucleoprotein called chromatin. Like this is interphase nucleus. Here we have that is non-distinct in the form of net-like structure, that is known as chromatin net. It is nucleoprotein made up of protein made up of that is nucleic acid, DNA, RNA and protein known as chromatin. But during stage of cell division, when the cell starts division, then this chromatin net that will divide into definite number of segments. Now these segments will be thicker and will be definite, and such structure is known as chromosome. Okay, cells are structural chromosomes instead of that is chromatin net. Chromatin, it contains DNA, basic proteins that is histone, some non-histone protein and also that is RNA. That's why known as nucleoprotein particle. You see a single human chromosome, that is we see here approximately that is has 2 meter long thread of DNA, and that is it will be distributed in 23 pair of chromosome. 2 meter long DNA is distributed in that is 26 chromosome. These chromatin fiber condensed to form chromosomes, in case of human being, that it will divide and it will give that is 46 such segments known as that is chromosome. Is it clear? Okay, come to the next now. That is this chromosome is differentiated into two regions, or we can say chromatin is differentiated into two regions. That is, one is euchromatin, one is heterochromatin. What do we mean by that? This is heterochromatin. It is darkly stained, densely packed area like this, while this is that is light stained and loosely packed area. This is the part of the chromatin. When this is lightly stained and loose, it is called as euchromatin. This is very active one. This is transcriptionally active, means here the transcription will take place. Here that is messenger RNA will be formed, while messenger RNA in time to come that will be responsible for that is protein synthesis, while in case of heterochromatin it is transcriptionally inactive. It doesn't make messenger RNA, right? It doesn't help in that is protein synthesis. Is it right? No protein synthesis. Function of nucleic acid, we can summarize: storehouse of genetic information, that is DNA. It's present in a nucleus, takes part in transmission of genetic information from parent to the offspring, that is daughter cell, from generation to generation. It controls all metabolic activities because it will make the enzymes which will control that is the activity of the cytoplasm. Division of the nucleus is prerequisite to the cell division, because when cell will divide, means nucleus has to divide. Okay, come to the next part now. That is, we have to say chromosome. Is it? Chromosome structure we have to see. I told you that this chromatin material of interphase stage that will divide into definite number of the segments, and these segments, these are known as that is chromosome. Each chromosome visible in dividing cell, not in undividing cell. This is interphase stage. Essentially has a primary construction or centromere. This is the primary construction of centromere. This one is centromere, that is on side of which a disc-shaped structure here. It is disc-shaped structure. That disc-shaped structure is known as that is kinetochore. That is known as that is kinetochore. And this central structure, that is, it is known as centromere. What is this? It is called, yes, it is called centromere. Okay. Based on position of the centromere, the chromosome can be classified into 4 types. You see here, the centromere is this one, right? And that is centromere had divided the chromosome into that is 2 halves. It will become like this. This is centromere. The 2 arms, this is one arm, this is the other arm. these are equal, then this is known as, that is, metacentric chromosome. You see here, one arm is smaller, one arm is larger, one arm is very small, other is larger, and here this arm, short arm, that is almost, this is absent, it is present at the terminal of this. So these are, that is, 4 type of chromosome. First I will give the detail, then we'll go back. Metacentric chromosome has middle centromere, as I told you here that the 2 equal arms are there. Submetacentric chromosome, slightly away from the centromere, it will be like this, that is, this will be V-shaped, that will be, what with shape of this, that will be, that is L-shaped. Acrocentric chromosome, that will be like this, that will be here, that is J-shaped, and telocentric chromosome, that will be I-shaped, that is has terminal centromere. Metacentric, submetacentric, acrocentric situated close to the end, and extremely short arm, and telocentric here. See, a few chromosomes may have additional, that is constriction, known as, that is non-staining part. This is important one, that is known as secondary constriction, or NOR, nuclear organizer, near the end. The part of the chromosome beyond the secondary constriction is known as satellite, important one, and a chromosome having satellite is called set chromosome, and these are considered as marker chromosome. Very important part this is. That this is the chromosome here, that is, this is the primary constriction. Some of the chromosome, they have secondary constriction here. Secondary constriction, also known as NOR. This very part of the chromosome beyond this secondary constriction is known as satellite. And what happens in satellite, that is here, the nucleolus will be formed, nucleolus organizing region, this is, and such chromosome which are having this satellite, these are known as set chromosome. Is it? In case of human, we have 5 pairs of set chromosome. You have to remember, 5 pairs of set chromosome we have. Is it clear? Go back. This is what? This is metacentric, this is submetacentric, this is acrocentric, and this one is telocentric, acrocentric, submetacentric, and that one is metacentric chromosome. Is it? Come to the next now. Now we have to say some special type of chromosome, also known as giant chromosome. Children, you see, in NCERT we do not have detail of this, but even then sometimes these are related to some specific functions. We have to see detail of this. Giant, these are large sized chromosome. We have to discuss 2 type of giant chromosome, special chromosome. One is lamp brush chromosome. The name is given, that is lamp brush. You know, that is to clean this chimney of lamp, that is, or milk bottle of the children, that is these brush like chromosomes, means brushes we used. Therefore, because of their appearance is like this, known as lamp brush chromosome. Primarily present in oocyte, very important point, oocyte of vertebrate as well as in some invertebrate. These are diploid bivalent chromosome, jointed at center point called chiasmata. The main axis, you know, that this is the main axis. This is the main axis, and this main axis, you see, that it is making this, that is loop-like structure. These are the main axis, loop-like structure. That is, these are made up of, that is lateral projections known as, that is loops. Main axis is made up of DNA. Each bivalent bear 2 rows of large chromosome. Loops are extended part of the chromosome. Very important point this is. And in transcription, that is this hair or loop, they actually, these are nascent RNA molecule. That is, here in loop we have, that is RNA, that is molecule we have. You know, RNA that will help in synthesis of protein. See, at the time of, that is development, when the zygote will be busy in so many work, to facilitate its work in advance, this messenger RNA have been synthesized, known as nascent, that is RNA molecule. These here are bound to protein, giving a fibril granular appearance, and some of these are stored as informosomes, important term, that is messenger RNA plus protein, for which is already well in advance they have been synthesized, and which will be used in future during development of embryo. Come to the next now, polytene chromosome, also known as salivary gland chromosome, because this is found in salivary gland of insects of order that is Diptera, first reported by that is 𝕍𝕀𝔻𝔼𝕆 in larval salivary gland of chironomous insect of that is order Diptera. In polytene chromosome, a process of polyteny occurs. Many chromatids are found per chromosome. You see that usually in chromosome arm there are two, but because of this polyteny, that is they do not separate after DNA duplication, that is in one part there will be that is many threads of chromatin, a total about that is 1024 to 2000 chromatids may appear in one chromosome. These chromosomes, they contain light band like this is the dark band, this one is the light band, in between the two dark band this is light band, that is, and the chromosome puff also called Bal-Biani rings. These are active center of what messenger RNA synthesis. Is it clear? That was part 6, in which we have discussed microbodies. Then we have seen detailed structure of nucleus, then chromatin, different type of chromosomes, and then in the last we have seen that is some special chromosome. Thank you.